Hopefully next time I can come physically be there, but with my teaching schedule this time, it just didn't work out. So at first I was going to talk about how to identify all different kinds of invertebrates that you'd see, but that's probably several days worth. And so then I thought, let's focus on the ones that are the background behind others in the photos. So I'm going for the small encrusting invertebrates. Let's see, I'm gonna pull up my screen here. Give me a moment to get this set. Oh, I think I need to swap that. There we go. All right. So encrusting invertebrates. So first of all, who am I? Hello, I'm Amanda Kahn. I study invertebrates. I'm a marine scientist at Moss Landing Marine Labs down in Monterey Bay. And I specifically focus on sponges, but I really broadly also have studied plenty of others. And I tend to focus on the ones that don't move around like this guy. I'll focus on the ones that are stuck in one spot and therefore have to interact with whatever comes their way. And so in this photo, if we take a look here, this is me, on this rock, we have examples of tons of different encrusting invertebrates. So we've got two different kinds of tunicates here. These look like bryozoans. This is a different bryozoan. Some cup corals here, which are cnidarians, and then algae and seaweeds, in addition to all these mobile urchins and, well, I guess that's all we see here. This was a little bit urchin barony, but all the mobile critters that move around. And I want to say, if you're interested, of course, guidebooks are fantastic, but there's also a great app that you can install on your phone from the Monterey Bay National Marine Sanctuary called Sea Photo. Or you can go to their website here and you can look up either a species or just browse their photo library and get an idea of what you may have seen in a photo that you have. And all the pictures are free to use in there. And many of the photos that are in my slides today come from here.
Amanda, could you sometime put that URL into the chat?
Yes, I can do that. I don't have it pastable right now, so I'll do that right after I finish the talk. If somebody could remind me.
Perfect.
Yeah, perfect. And I can send a little summary too. So as an invertebrate zoologist, I study a few different groups. Actually, there are 32 different groups of animals that exist. And this is sort of a family tree that shows the relationships of all of these different phyla of animals that we have. And of the approximately 1.38 million species that we currently know of, 96% of them are invertebrates. So it's a lot to cover. And we're not going to talk about all of them. We're going to talk about the ones that don't move much. And I don't know how long it's going to take to go through them. Oh yeah, since we started late, when should I aim to stop? Because this is a very flexible kind of talk. Is there a set time?
Yes, Amanda, we'd appreciate
30 to 45 minutes.
Right. If you would,
okay.
If you would finish at 10 past, and then we'll have 5 minutes of questions.
Okay, sounds good. 10 past. Okay, great, thanks. So then I'll talk about a couple of these groups, 2, 3, or maybe 4 if we have time for it. And so I'll introduce them a little bit, like who are they? So here are the sponges. I will talk for sure about the bryozoans here, possibly about the chordates. Where are they? Oh, chordates. Well, there we see, we're chordates too, but I'll talk about the urochordates or the tunicates. And then if there's time, maybe some other groups as well. But you can see that there's a lot of other diversity that's out there, which I think is just fantastic. So let's dive right in, starting with my favorites, the sponges. And for those of you who saw my presentation last year, you might recognize a little bit and know a little bit about these guys. Sponges are filter feeders. They draw water in through their body wall, and then they filter particles out from the water, especially bacteria, before they pass that water out of their body as you can see here with that non-toxic green dye that we use. And they actually, you know, when you look at a sponge, it might look like a lump on the seafloor, but actually they're constantly pumping water. And if you take a look at that little sponge in the right-hand corner, this is a time lapse of images, and you can see that actually sponges are quite active, and they'll change their shape, their size, and whether their pumping units are open or closed, depending on water currents or other things that like irritants. So they're pretty cool dynamic creatures, and they build their skeletons out of these small glass rods. The glass rods could actually be lots of different kinds of shapes, but what that means is that sponges tend to look, well, kind of amorphous or spongy, and they feel a little bit rough to the touch. And if you were to break one a little bit or rub your finger against it, it would feel rough. It wouldn't be smooth or rubbery. And that's in contrast to one of the groups that you'll see later. This is a good touch test to recognize the different groups. So who are some of our common sponges in the area? Well, this one might be the most identifiable or recognizable. Does anybody know this one? This is the red volcano sponge. And I put the scientific name down as well in case anyone wants to look it up in different ways. Okay, I'm gonna get my video up so I can see you all too. Okay, here we go. Okay, so the red volcano sponge tends to grow in places like the shale beds off of Del Monte Beach, where it's sort of a sandstone type habitat. But they also grow in inner Monterey Bay a lot, in the bay. I don't see them as dominant in Carmel side or area, but you know, your results may vary. So they are recognizable by having these pores called oscula that are raised up on little volcanoes and a bright red color. The other red sponges that we have in the area don't have those volcanoes.
Amanda, sorry to interrupt you. Can you suppress your header on the screen or give it full screen view so we can actually see the good stuff at the top?
That's interesting that that's visible. Yeah, how do I hide it? Hide. There. Is that right?
Yay.
Yes.
Oh, thank goodness.
Oh my gosh, you solved it. All right. So next sponge, this one. You may have recognized this as just blue rocks that you've seen when you've been out diving, especially when you shine a light on it, but this actually isn't the color of the rock. This is a really, really thin sponge. So whereas the red volcano sponge is maybe as thick as your finger as it grows on the rocks, this sponge, the blue cobalt sponge, is extremely thin, less than 1/4 of an inch. And so it will basically look like just a blue rock that forms. This guy is the orange puffball sponge, Tethia californiana. These have some pretty strong behaviors. And so if you were to set out a camera at the beginning of a dive and leave it there watching this sponge, you would see it contracting and pumping its body into bigger and smaller sizes as you look, which means you might see it with these little islands with gaps and spaces in between, or it might be all shrunken down and with those islands squeezed together and look like a solid ball. This guy is one of my favorites because it's basically looks like a gray tennis ball. So these guys, Craniella arb, the gray tennis ball, these are, I never have seen them inside of Monterey Bay, but you can find them in Carmel at Monastery or Carmel River Beach or Butterfly House. So from that, we expect that they like more water flow and kind of, you know, more open water environments. We don't know a lot about a lot of these sponge groups. That's part of my goal in my research. But right now we're sort of just documenting who's found and where. And I do want to say from all of these pictures, you can see there's a bunch of other encrusting life in the background. And so part of the fun that I have when I look at a photo now is I'll look at the main critter, and then I'll also ask myself who these others are in the background. And I don't know many of those, but this here, that's a tunicate, right? These are cup corals. We've got some bryozoans back here and then some other species of sponges. So yeah, quite a bit in there. This one is another one that I mostly see, you'll see in Carmel area. So this is just the only brown sponge that we really have. And it's pretty thick and fingery. And you can see this one really shows that texture where it would feel rough to the touch if you touched it. And then you also have some smaller, cute little guys. So these guys, these are small vase sponges. They're seasonal, so you'll see them sort of sprout up, and you might see one or two this time of year. And then once upwelling happens, you'll see clusters that form where the sprouts are. So they're all connected at the base like a little stand of trees. And then this one is a frilled sponge, Leucoselenia eleanor, and it can grow as these tiny, tiny tubes that are either flat or that make an orb or ball. And we've been studying this one to understand how it pumps water through its body. So I have a video I'll show where we use that non-toxic green dye as a tracer in the water. That dye is actually the same as what makes Mountain Dew green. So here is Leucoselenia there. And this one is ball shaped, and we inject the dye near it, and then we wait and watch while the surge blows the sponge back and forth, and we also see how the green dye moves around it. So this one is kind of funky. I have some videos that I'll show later that show pumping through things that make a little bit more sense of how the dye moves. Okay, so those are the sponges. There are a ton more, and I should say if you want to see more sponges, Sea Photo has a listing of many, many more, but hopefully some of those look familiar to some of you as ones that you've spotted and weren't quite sure what they were, or maybe just passed over or thought that it was a very colorful rock, and now you know it's a sponge. And now I want to go to things that I used to always mistake as sponges when I first would see them, and those are the tunicates. So to put on my classroom hat for a second and introduce you to the tunicates, there are about 3,000 species, and they include the tunicates that we see living on the seafloor, but also they include salps, which float in the water column and which you might see while out diving. And pyrosomes, which we've also been seeing diving lately in the last few years. But to talk about the ones that live on the seafloor, they basically work by kind of like a sponge, drawing water in through a siphon, filtering particles from the water on this part of their gills before passing the water back out of another siphon. But the neat thing is, tunicates can either live solo like this, or they can clone themselves and make themselves either kind of grow like strawberry plants where they have these stolons that connect individual clones of each other, or they can actually have a shared common base that they all sprout from, in which case they actually share nutrients as well. And so it makes it a really fun and interesting puzzle to ask, you know, what makes up an individual here? Is each individual zooid its own individual, or is this a cluster of clones? Is each one a different individual? What about these? And then it gets even weirder because these clones, at least they all are distinct by still having one water intake and then one siphon that lets water out. But you also have tunicates that can grow as compound tunicates. And for those, they form these clusters like these ones that you see here in close up. And these compound tunicates, each individual clone takes water in, but then it pumps the water out of a shared common exhalant spot. So they basically share the water outgoing passage, but they all have separate ones that go in. It means that they get these beautiful patterns, and all of these clones now live embedded inside of a tunic, hence the name tunicates. So all of these, whether they're solitary, colonial, or compound, they are surrounded by this tunic material that makes them feel rubbery to the touch. When you touch a tunicate, it's going to feel kind of springy. I kind of think they feel like a hard-boiled egg. So if you touch it and it's rough, it might be a sponge. If you touch it and it's rubbery or smooth, it's more likely a tunicate. And so that's a nice way to take a look at one of these compound guys and recognize them as a tunicate. The other way that you might be able to tell is because if you look close up, you can see all of those individual clusters or systems that have those shared chimneys in the middle. So this one, it's not circular, but there's one system with lots of inflowing pores, but only one chimney. And here's another one with one chimney. So this whole thing is a colony of clones, and each of these little dots is one individual that makes up part of that colony. They're all embedded in that shared tunic. So this one is called a Aplydium. It kind of feels like Spam or bologna, and it grows thickly, like maybe an inch or so thick. And this one responds to touch, which is kind of interesting. So when you touch it, it'll close up all of those pores and shrink down. But let's look here. This is an example of a colonial one, right? So not a compound. This one is the light bulb tunicate, Clavolina huntsmani. And you can see each one of these individuals has an in and an outgoing water siphon. So each one is an individual, but down here, they grow one clone after another. And you can see some baby ones growing up here, some baby clones. And these are called light bulb tunicates because, as you can see, they have these filaments inside that look like the tungsten filaments of an old incandescent light. Another tunicate that we have a lot of down here is, this one is a compound tunicate. This is the lobed tunicate. So they'll grow all sorts of shapes and colors. So here's an orange, a white, and a purple. These guys are really, really good hiding spots for cone snails, conus. So if you go in here, you might see cone snails nestled in. Also chestnut cowries at Monastery. This is like the way that we can reliably find chestnut cowries is if we look inside of these lobed tunicates in all the folds. So they make great habitat, and it's neat to start recognizing the sort of pairings and groupings that the different species make. Okay, here we go. This one is a tiny version of a tunicate that can also grow as full sheets. So they either can grow separately as little tiny individual compounds like this, or they might grow as big sheets that flow together. I always thought these were sponges, so these are a little bit tricky. They feel a little bit more crunchy than a typical tunicate, not as rubbery. And you'll see a lot of this growing over other things. So it's really a good competitor for space. And here it looks like actually there might be a war of different clones going on, because you can see that this tunicate is growing over and around this more pale orange tunicate. So we have our darker orange and a lighter orange there. So these guys, you can really, once they start growing, can really take over. These are also eaten by a lot of snails. And so the top snails, like the Caliostomas, these top snails will often be seen grazing on various types of tunicates. And then you might also see some of our solitary tunicates as well. So these never grow in clusters or groups. You'll see just one. This one is, I don't think it has a common name, but it's Styella, which I always remember because I think of A Streetcar Named Desire, right? Styella. And I get excited when I see it. These guys are neat because they grow on a stalk, and they look kind of reddish brown and leathery with grooves. And here's another view of one from the side. And they can grow up to about 5-6 inches long, and so they can be quite large. These ones will flop around in the water. And so as they flop from side to side, as the surge moves, their inhalant siphon actually orients so that water pushes its way inside of the inhalant siphon. So here I have another video of where we were adding that dye to see the water flow, and you can see the way that as the surge moves it, it pushes more dye out of the tunicate when the surge is strongest. And then when the slack comes, it pumps less water until the tunicate kind of gets pulled in the other direction. So yeah, that little siphon there flops over and water can get pushed in there and help these guys filter feed. So these guys, maybe big, I don't know whether they grow there or whether they just survive better, but they're all often found among red algae, which they blend in with quite well. But you'll see them flopping around. And then this is another one that doesn't have a common name, but I call it the peach tunicate because I think it looks like a nectarine or a peach, maybe more like a nectarine. It doesn't have the fuzz. But these guys are very distinctive. Sometimes they'll look like a nudibranch, especially if you touch them and they pull these siphons back, and then they just look like a sad little flat pink sheet until they sort of inflate themselves back up. But they're glossy pink to a pale red with two really apparent siphons that stick out, and they filter the water. And finally, I don't know who's seen this guy. I remember seeing this and not knowing if it was a plant or an animal or kind of what it was, but this guy is the orange club tunicate, and it has spikes all over its body. So it looks like an old medieval mace. It's actually pretty common too. Once I started seeing them, it's easy to find these guys all over the place. They're just rather short, so they're hard to spot initially. So also in this group of tunicates are a few representatives that show up in the water column that you might spot as well. So pyrosomes are one of those groups. They're also called sea pickles. And these guys are basically tunicates that don't live on the seafloor anymore. Instead, all of those little individual clones live inside of a tunic in a tube that drifts through the water. And so as those little individuals draw water through their bodies, they pump it out through a single hole in the backside of the animal. And that creates a water jet that helps them swim through the water. And so you'll see pyrosomes sometimes drifting through the water. And we have ended up since 2013 with massive blooms of these pyrosomes. Sometimes they'll wash up on beaches in big numbers. And with my work in the deep sea, sometimes we'll see them grow en masse and then sink to the deep sea floor. And then the other group that is also a tunicate that swims is the salps. And so salp chains form basically kind of like those colonial tunicates do on the seafloor, where they have individuals that are connected to each other and make big long chains or rings that are all clones of each other. So that's all the tunicates I'm going to talk about. Does anybody have any questions or extras that they want to share of things that they've seen or things that they weren't sure what type of animal it was? Should we go to the next group? Yeah, Arnie?
Well, it's a weird question. You mentioned that when you touch some of these, they contract. Yeah. Now, eventually they return. Is there a constant time interval that they use?
A constant what?
They're not looking at anything.
Oh, yeah. I don't know if there's a time that they wait, but these guys, they have a nervous system. So if they feel something, it's usually by sensing a change in the flow of water around them. So, you know, that's why even if they can't see, they might respond to you approaching to touch. And then once you touch, if the water remains unchanged, you know, it seems like you're gone, they'll open back up. But I don't know if there's a specific time that they wait before they start.
So
yeah.
Amanda, that brings up something I wanted to ask about the tunicates. My understanding is they're about the most primitive, at least in the sea, the most primitive life form that's got a notochord or you know, the beginnings of what would makes them like us. So can you tell us about the development? How do these things, presumably it's a larval stage, they come from eggs, and then did they always have, as far as we know, always have that notochord, or how does it develop?
Yeah. Oh, that's a great question. Yeah. So let's see. Am I able to draw on my slide? Looks like not. That's okay. I'll draw with my mouse. So so yeah, while tunicates look something like what you see here once they're adults, when they're larvae, they have a head and a tail. And that's what the larva looks like. And then in the larva, they have a few key features. So they have that head, which since I can't write the whole words easily, I'm just going to say H. They have a tail. And then, yeah, they have the nodal cord, which is a stiffened rod that runs right next to a dorsal nerve cord. Now, these might not sound like they're unique or special, but actually they are. So in many other invertebrate groups, before we get to these guys, the nerve cord runs along their belly on the ventral side, so down here. And so it's actually different and unique to have it back here. And if you think about us humans, we're chordates. We have our nerve cord in the dorsal side. And so this is the first group that got that development. And that nodal cord, that stiff rod, that helps to protect and give structure to the tail. The other things that make these guys unique is they have gills that form right behind their pharynx. And we actually have the same thing happen during our development as well, which is like super frustrating as a diver to learn that during our development, when we were tiny, tiny embryos, we had gills and then they closed up and we kept on going and developed lungs and all of that and had to get ourselves back to being able to breathe underwater. So once the larvae swims along, it'll settle down, it'll find a rock somewhere that it's going to grow onto. And when it settles, it actually glues its head to the rock, and the tail with its nodal cord and its dorsal nerve cord melts away. And so what you're left with is mainly just this head. And you have a little bit of nerve cord. You have a bit of a stiffening here from the nodal cord. And then you also have those gills remain, and they turn into what in one of my previous pictures is now this area that pumps water through the body and filters water. So this pharyngeal basket, it comes from those original gills. Yeah. Does that kind of answer? That's a bit of how they form from something that otherwise we have a larval stage, we humans, that's very similar to that. And if we look at where the tunicates sit in relation to us, going back to this giant family tree, the tunicates are here and we are craniates. So we are here. So they're our last invertebrate ancestor that branched off from our shared common ancestor that had that nerve cord.
Yeah, that's fascinating, Amanda. Thank you. I'm sure we could spend a whole hour just talking about tunicates.
Yeah.
I like that. Thank you.
Yeah, you're welcome. Oh man, I definitely do spend hours talking about these. So I hope it's not boring that I just go on, but man, I just think they're the most alien things ever to think about. You know, when we look at a fish or a whale or a human, right? We kind of know already that they've got a head end, they've got a tail, or they've got limbs. They might have nerves or a gut that starts at one end and ends at another end. And then you go to an invertebrate. I mean, and you think of a sea anemone, and they take food in through the same hole that they put food out. So their gut doesn't go all the way through like ours. And they actually don't have a head. But why do they need a head? And when we think about something like a tunicate, they give up their head after their larvae, right? Like they glue themselves to the seafloor. And once they're glued to the seafloor and pumping water through their bodies, where would a head matter here? And so it ends up that you end up with these bizarre shapes and styles and ways that life can exist, which I just think is so liberating. Man, we've got some real aliens living on this planet. Does anybody have more questions before, or should we move on to another group?
I have a question. Going back to your earlier slides, you showed these long needles with the sponges.
Yeah.
Are those silicates?
Yes, they are. Oh, great observation. Yeah, so sponges actually take silica and oxygen from the seawater and they deposit it as glass. So these are opaline, amorphous opaline or opal, which is silicate basically, or silica based.
And tunicates then are all carbon based. All their structural material is based on. Oh, okay, that's a good differentiation then.
Yeah, and so a tunicate will never feel crispy or crunchy, with one exception, whereas sponges almost all will feel quite crunchy. The one exception of a tunicate that might feel a little bit crunchy is this didymnum. They make calcium, small tiny calcium nodules inside of their tissue, which helps to resist some of the grazing by those snails that come by and like to eat it. So they have a little bit of defense.
When looking at a particular genus or anything, you ever use DNA comparisons to do, just you do morphology, obviously, but do you ever do DNA comparisons?
Yes, definitely. And sometimes we have to. So, you know, it's even looking at this photo, I would have a hard time saying which particular species of didymnum this is. And so sometimes for studying them, we really do have to take a piece and look at the DNA to figure out what species it really is. And for the sponges, there's a researcher at UC Santa Barbara. Ooh, he'd be great to give a talk to the group too. And he's been cataloging photos and DNA of all the sponges up and down the coast of California. And actually he logs them in iNaturalist, so you can check out his photos and see some of his work there. I'll share his info after as well.
Great, thank you.
Thanks. That was a great question, Gary. And if all these other interesting sites, links, people, as long as you can send them up to us, that would be great afterwards. Let's let you get back to the talk, and we'll have some more questions at the end.
Okay, sounds good. Yeah, so I do want to talk about my other favorite group, the bryozoans. I feel like they don't get a lot of attention because they're quite flat and quite small, but actually they're microscopic and awesome. If you take a look here of a side view, so maybe we'll look here. This is what some bryozoans look like. They'll either grow up in these sort of flutes and leaves, or they'll grow as these very beautiful flat geometric grids and patterns, or they'll grow as stalks with little critters or individual clones growing off of those stalks. Collectively, they're called the bryozoa, which translates from Latin to mean the moss animals. And they used to also be called fairy lace, which I think is just lovely. But if you look close up at any of these, whether they're these very rectangular geometric ones, or these growing off in stalks, or these, if you look close up, each little chamber is an individual clone again. And those clones are each filter feeders. And so here you can see the little fingers or digits of those bryozoans being attracted to phytoplankton or algae cells in the water, and they're actually taking them, and whenever they feel an algae cell touch one of those tentacles, they whack it toward the mouth, and so it looks like kind of like hungry hippos, right, where it sort of just bats it. So they're busy. There are about 4,500 species, and they live in the ocean and in fresh water, so you can find freshwater bryozoans. And you don't really, well, maybe we just don't see them, but usually you don't find just one individual. They grow as these big colonies. And here is probably the easiest one to spot that, you know, the original bryozoan that settled from a larva was probably right about there. And then it started cloning itself and grew and grew and grew until you see the outer edge that's still growing here. And these guys can have certain individuals that are, while all clones, they can become specialized to either be for feeding or reproduction or protection of the whole colony. And they look, well, something like this. So they can be very flat. They're often brittle with a rough texture. So kind of like a sponge, if you rub it, it's not going to be smooth. It'll be more grainy, but unlike a sponge, it won't be spongy. And so these guys are at the further end of being more crunchy, sponges being more rough, but a little softer, and then tunicates being that sort of hard-boiled egg and rubbery feel. So here's another bryozoan up here next to that didymnum tunicate that I showed before. So you can see that they also can grow and take over a lot of space on the rocks. And once they grow, it's hard for other animals to grow on top of them. And so that's why they can take over large portions of the rock. And even this one is growing up the side of that cup coral. And you can see here, what I like about this is you can actually see a little bit of fuzziness to this bryozoan. So that fuzziness is all of those little tiny individual clones sticking their feeding appendages out to catch food. So there it is with all those feeding appendages pulled back. There it is with them sticking out. So a few of the ones you might see around Monterey Bay, this one, the fluted bryozoan, looks like a pile of cornflakes. And often with this one, you'll see snails and flatworms embedded inside of the flutes and piles of cornflakes. These guys grow pretty large, you know, a few inches in diameter, they can be. And then we also have the staghorn, the southern staghorn bryozoan, Diaperoforma californica, which look kind of like a little branchy coral. You know, they're sort of an orangey yellow color. They have these branches that sort of split and then split again. So here they have a Y shape that then splits again into another Y. And these are really a neat group because, let's see, is this the picture? Do I have my, oh, it's the next species. So this group can have a lot of other animals growing and living inside of it. But the one that I wanted to really highlight is the northern staghorn bryozoan. And this guy, it looks similar to the southern, but it's got thicker branches, and those branches, while yellow at the tips, are sometimes more of a purple or reddish color further down. And the cool part about this is it often houses tiny purple ring top snails. And so we think that this might be a nursery for those top snails to grow on, because usually the ones that we see are less than a centimeter long. And so they're really quite small and quite young. Probably, who knows if they're eating them. It would be a really neat study to figure out if that's what they're doing. This one is one of the prettiest, I think. This is kind of like the cornflake one, but it's a lacy cornflake. So this is the lacy bryozoan, Phytolopera pacifica. It can either be really brilliant orange color with these holes, or a little bit more of a pale orange, faded, or with some brown in it as well. These grow more on the Monterey Bay side than the Carmel side, but I have seen them in smaller colonies in Carmel Bay quite a bit. And then we have one invasive species of bryozoan. We probably have several, but this species, Water Cypera, is quite abundant now within Monterey Bay and Carmel. And so it's this dark red bryozoan. And if you look close up, this is great, has plenty of critters living on it. But it actually reminds me, when I look close up, of a watermelon, because you can see that at each individual clone, you have a black operculum or trap door that covers those clones. And so, you know, these guys basically look like they are a red bryozoan with these black watermelon seeds throughout. And these can grow either as flat crusts if the water is moving really swiftly, like you can see back here, or if the water is more calm or still, or if they just grow too much, they'll start growing up and folding into these beautiful folds. It's actually a really lovely bryozoan. It's just too bad that it's invasive. And we don't know all of the interactions that happen with it, but it seems to be integrated quite well. These guys can grow on the copper-based paints that are used on boats for anti-fouling. So that's part of why they're really successful invaders. And then this is another bryozoan that you're likely to see while out diving. Hello, I love all these nudibranch cameos. There's a little nudibranch hiding right there. But this is Membraniptera villosa. So it grows specifically on brown algae, like this giant kelp blade, or like some of the laminaria, the sort of shorter kelps that grow. And you'll see each of these rounds was probably a single individual larva that then grew up into a colony. I don't think this picture this picture may well have one, but when you're doing a safety stop, it's really fun to find Membraniptera and to look close up to find nudibranchs living on it. You'll see this guy, and unfortunately, I don't know that guy's species, but there's also a very flat nudibranch that essentially looks like this. It's white with brown spots on it, and it just blends so perfectly. And it's a specialist that grazes on Membraniptera. So it's a really fun little ecosystem that you can find while you're waiting on your safety stop. And then this one is one that grows a little thicker on red algae in shallower water, maybe 15 feet to 30 feet. So this is Ferrucella brevituba. It doesn't have a common name, but I think it's a really recognizable one because it grows over red algae and it feels very leathery, unlike the crunchy other bryozoans that we have. And it grows a lot on the chain bladder kelp, especially. And then this one I included because I always think it looks like a pile of Cheetos on the seafloor. Maybe I'm just hungry all the time during dives. So the Cheeto bryozoan here is thick and chunky and, you know, just lovely because it makes me think of my post-dive snack. So these guys are bright orange, and honestly, it's the best description. They're like Cheetos. And so those are some of the common bryozoans that probably you've seen on your dives as well. And looking at the time, I think I won't go into more groups, though I have tons. I love them. But I hope that now when you look at, you know, a picture like this, you'll see, ooh, there's a bryozoan taking over from those, you know, from those cnidarians. And here we've got some encrusting algae, but there's a bryozoan as well. We've got a grazer here. Hello. We've got, well, we might have a little bit of sponge up here. We'd have to look a little closer or maybe touch it to find out. But yeah, you know, when you take your photos now, take a look at what, you know, what's in the background as well. And I think it's really fun to see who's there and who's growing over whom. And yeah, with that, I'm going to stop showing more pictures and take some questions and make one announcement, which is that if you like this and you want to learn more about fun invertebrates and marine life, my marine lab, Moss Landing Marine Labs, is going to have an open house. Right now, I don't have a flyer for it, but I do have a save the date that I can share. Oh, I'm trying to stop sharing screen, but I can't see it anymore. Let's see. Unshare. Gosh. I can't see how to stop sharing my screens because I hid the video controls, which is awkward. I'm sorry. I'll figure that out. But yeah, I don't have a flyer to show you.
So select the view options and change. There we go. I think you're okay.
Well, that's my cat, but yeah. Well, we'll figure it out. Oh, here we go. Yes, got it. There we go. We're gonna have an open house April 30th. And so it's one day, it's all outdoors, and we're gonna have a touch tank, and we're gonna have a bunch of marine science students who take a whole class where basically we do this, and then we go out and we explore and talk about all those different interactions of who eats whom and where to find the baby snails and everything else. So come and grill us or tell us some of your observations, and you might inspire a master's student to want to go do a whole thesis studying something that you've just been noticing as you've been out diving, because honestly, you are the eyes watching the ocean. So yeah, I'll share information with the club once we get a handout for it, and I'm happy to take questions. Oh, I see Helga has a question. Since the urchins are running out of food, will they start cannibalizing the sponges and tunicates? Oh, you know, that's a good question. I think they're herbivores, and they've never really targeted non-algae as food. I'm not sure actually if they're capable of digesting it, but definitely they don't seem to be grazing down the sponges and tunicates, because in the urchin barrens in Carmel Bay, we still see tunicates and sponges growing in those barrens. But it would be interesting to look at that and ask whether, you know, we see any sponge skeleton pieces inside the stomachs of the urchins. I wish I had a better answer. We don't know.
Online, if you want to ask a question, either just speak up or stick your virtual hand up, and then here in the room, if anybody here, Gary wants to ask a question.
Hello. I've done some work up in Alaska, and the Inuit up there eat tunicates, and they'll eat 3 species: the sea potato, the sea peach, and little ones called sea grapes. And I've actually tried it. So when they're raw, they taste like gushy seawater. When they boiled them, they actually were a little bitter.
Huh.
Have you ever?
I have never heard of that. That is, wow. I want to look that up and learn about that. One thing I remember being taught that's strange about the tunic of the tunicates is that it doesn't have cells inside of it. It's like it's organic, it's made by the animal, but it's not living. And actually it has a high concentration of vanadium, which is an element somewhere in the periodic table quite high that isn't very concentrated anywhere else, but for some reason it concentrates in the tunic of tunicates. And so I wonder whether the Inuit either, needed more vanadium in their diets and so naturally gravitated toward a source, or if you just got a high dose of of something.
The way they harvest them, they would just take a rake and just scrape it off the bottom.
Oh, wow. So I had a lot of medically important compounds come from marine animals as well. So sponges are one of the top sources. So are marine worms. Tunicates haven't been explored as much, but we extract several drugs for anti-cancer, antibacterial, or antivirals from marine life. And so I wonder whether maybe that bitter taste or anything else could come from those compounds.
Well, I know that molybdenum is harvested from the sea, and maybe vanadium. You make chrome steels and things use vanadium and molybdenum. So maybe the steel industry will be interested in tunicates.
That's more sustainable than where they get their molybdenum from, right? So deep sea mining is where they've been exploring for that. So if we could convince them to stay away from the deep sea nodules and go after farming tunicates instead, that could be pretty good.
Anybody else with a question? Devin.
What are the sponges that are used for sponges?
They're a specific, very small group of sponges that happen not to make those glass skeletons. So they only have protein called spongin. And so when you, I have a slide in another talk. When you pull them out, they look like this black sludge. And then when you bleach them or clean them off, all you get left is that spongin. And it honestly looks just like a bath sponge. So yeah, they grow them in Greece, and then also in America, a Greek community brought over their sponges and grows up a few of the species in Tarpon Springs, Florida. So if you ever go out there, you can dive in some of the sponge farms. Love it.
Thank you.
Yeah.
And that's quite famous over there. There's a big Greek community that would go over to that. Anybody else or anybody online? James.
Yes, actually.
Yeah. I had a question about the bryozoans at WRF2, which I think you showed a photo of that they're apparently an invasive species. And what a lot of us have noticed about them, though, you showed some nudibranchs, but a lot of things seem to hide in them. I mean, I've seen crabs, I've seen fish, and all kinds of stuff. So I guess what my question is yeah, they're not native. Are they harmful? I mean, are they crowding out native species or causing a problem? Because they shelter other animals. So.
Yeah. Ooh, that's a great question. I think that it's all a continuum of, are they good or are they a problem? These, while they're pretty good at invading, they have not spread quickly, and they compete with and sometimes win, but don't totally overtake their environment. In contrast, Sargassum down in Southern California, Sargassum horneri, you would say that's a pretty bad invader. So yeah, it's true. It's not one it doesn't seem to be doing a terrible thing, right? It's not overtaking everything. And it is having some interactions that aren't bad, things are eating them. Those nudibranchs were laying eggs on them. And so, yeah, it's a hard question. I feel like it's all a judgment of each species by species basis, whether they're having a good, a neutral, or a bad effect on them.
Debbie, did you have a question?
I did, and I was thinking it's kind of off subject, but I was thinking of, I know they call them mermaid's pearls, and that made me think when Gary mentioned the sea grapes, if that's similar to a mermaid's pearl and it might be a tunicate. Do you know what I'm talking about, Amanda?
I do, I think. Is it mermaid's purse?
No, pearl.
Oh, pearl.
Mermaid's
Okay, I don't.
purse is the skate, or the, you know, the.
It's
the
yeah,
skate.
it's the skate egg place. Okay, I'm Googling mermaid's pearl because I don't know that. And maybe I need to look up marine. Sorry, I'm not sure exactly what they are. If you could send a picture of one.
I'll have to find one.
Try, yeah.
Because it just made me think of it. I was trying to imagine what Gary's, when he was talking about the sea grapes, what that was and what that looked like.
Yeah, and I'm curious about that too. Which species the sea grapes were, because I think the sea peach and the sea potato, I can picture which tunicates those probably were, but I'm not sure for the sea grapes. Yeah.
I'll describe it. It looked like a branch with white tunicates all spread around.
Oh, interesting.
Those were small. The sea potato and sea peaches are about the size of a potato. But the sea grapes were like a big grape, but clustered on a branch.
Oh, okay. Were they something like this guy? This is small, and little clusters at the top. Or else one of these. Yeah.
No, it looked like a branch with white tunicates on.
Okay. Huh.
We're kind of out of time, so I'm going to suggest that Gary, maybe you'll contact Amanda offline and you can go and research sea potatoes and sea grapes. I must say one of my favorite things to see in the ocean is sea pearls, which are just these huge single cell creatures, and they're quite remarkable.
Oh.
You haven't seen them? They look like a glass bead, and they're huge. They're single cells, but they have a very tough skin, and they're as big as maybe half an inch.
Oh my
You know,
God.
I think that sea pearls were just lead shot.
No, they're not lead shot. Although that's a problem in itself. Anyway, Amanda, clearly there is lots of interest in these invertebrates, and we're going to have you back in another 6 months or a year to give us more of this. I think it was very popular, and I'd like everybody to thank Amanda with a either real or virtual applause.
Thanks, everyone. I'll see you at the club dive. I'm going to join you.
If it's very quick.
I find this very fascinating. I'm going to make a suggestion for you to expand your research. Why don't you figure out which ones are the tasty ones?
I'm going to need a lot of test subjects for that. Thanks. I'll make sure to reach out.
I think you have a volunteer. Gary's going to taste everything. Or both Garys. Okay, thank you very much, Amanda. If you would like to hang up, hopefully next time you can actually come and be here, and that way you get free pizza. But if you stick around, or you're welcome to stick around. If you need to go, we'll say goodbye, and but do come along to the club, and we'd like to dive with you. And if you give us the information, we'll put it up so that people can come along to the open day. We'd like that. Okay. Thank you. Yes, Amanda is always was very enthusiastic. Still is. Thank you once again. Okay, let's move on to our regular agenda. And I have actually sent it out, but I have to look it up. But I know the next things are the director's reports. So being pro tem, the entertainment director, I'll report on that, which is that we had Amanda tonight. And one other thing, Amanda, please, if you wish to share your slides, we talked about that, or give us more, a write-up. I know we got your bio. We can put that in the newsletter or in our archive, And then we have next month, I do urge everybody to come